We are the Tardigrade trio. You can tell because we have 8 legs and a segmented body.
We live on both land and in the water, but we're well known for our ability to survive conditions in complete opposition to our natural environment, including the vacuum of space.
We've been around for over 600 million years, so we're more ancient than the dinosaurs. We're here to share all the wisdom we've collected over the years with you.
Let's begin with a fun fact. Did you know that approximately 1/6 of the tardigrade genome is foreign? This makes us the animal with the most foreign DNA ever.
Yeah, the previous record holders, rotifers, held the record at up to 9%.
Whoa, that's amazing. But where does this DNA come from?
Well, originally it was thought that we borrowed genes from different bacteria, but actually we use a mechanism called horizontal gene transfer. This basically refers to the movement of genetic material between organisms that is not inherited. Essentially, tardigrades are thieves that steal genetic material from other species, like plants, bacteria, fungi, and archaea.
That's crazy. How exactly did we manage to steal so much foreign DNA?
The exact method still remains unknown, but is suspected to be the result of one of our survival mechanisms called anhydrobiosis.
Wait, how does that work?
Anhydrobiosis is a form that tardigrades can enter when there is no water present. It's a dehydrated state called a ton that is ametabolic, so the rate of metabolism decreases to a near undetectable level. With this, we achieve equilibrium with dry air and also reverse it by rehydrating into a highly stable active state.
So essentially, when we dry out, our DNA breaks into fragments, and once we rehydrate, our nucleus remains leaky for a while as we try to fix ourselves, and sometimes we may accidentally acquire other DNA.
Whoa, that's so cool. But how do we know what's foreign and what's not?
Well, I'm glad you asked. Let's take a dive into the tree of life to find out. Previously, many scientists and researchers believed that tardigrades were descendants of arthropods because early family trees were based on anatomy, and many organisms with segmented bodies and legs were linked together.
Like insects and spiders?
Yep.
And also crustaceans, trilobites, and tardigrades. These were called panarthropods.
So tardigrades are actually descendants of panarthropods?
Yep, and we're actually more closely related to the nematode and nematomorphs than arthropods.
No way. Roundworms and horsehair worms? How's that even possible?
Well, the secret lies in the HOX genes that regulate the development in all parts of the body. Most bilateral animals have 10 HOX genes, but tardigrades, nematodes, and nematomorphs have only 5, with the same 5 genes missing.
Who would have thought? So how does this help us?
Well, by understanding our origin, we may be able to completely trace the path of our DNA acquisition and determine where this DNA comes from.
Then we can use this to see which genes help us to survive in extreme conditions.
Extreme conditions such as temperature, pressures, and radiation?
Yep. One of the main mechanisms is what we mentioned before, anhydrobiosis.
Ooh. How does that help with temperature resilience?
Well, we basically dominate the entire world, since we can live in both super cold places like Antarctica and super hot places like deserts. We have a wide tolerance that ranges from negative 200 degrees Celsius to 151 degrees Celsius, but unfortunately, we can only survive at these extremes for a short period of time.
But we can survive up to 30 years frozen and up to 10 years in desiccation.
That's such a long time. Which genome mechanism helps with this?
Well, we can synthesize a bunch of special molecules called trehalose.
Oh, like the disaccharide that increases the denaturation midpoint temperature of proteins?
Yep, and so these trehalose are like little armies that protect our cells whilst we're in anhydrobiosis.
But I heard somewhere that tardigrades only accumulate limited trehalose whilst in anhydrobiosis. So there must also be other heat protection mechanisms that protect the cells and DNA from extreme temperatures, right?
Yep, there are.
Ever heard of CAS? It stands for Cytoplasmic Abundant Heat Soluble, and it's a tardigrade unique protein that is also duplicated greatly in the genome.
That sounds like a really complicated protein, and hard to pronounce too.
But it's really important since it protects our cellular components from surrounding temperatures whilst we are in anhydrobiosis.
It can also extend its protection to cell membranes, so it can effectively protect against temperature fluctuations of seasons in the long term too.
Yeah, Earth's temperature right now is so unstable, especially with the whole global warming thing going on. I've always dreamed of moving to the moon in the future. I feel like that's where all the fun is.
But remember, we are only capable of surviving up to 10 days in outer space. But I'm sure in the future, with improved technology and advancements, you can make that dream of yours come true.
Oh man, I thought that since we can survive in pressures ranging from the vacuum of space to 6,000 standard atmospheric pressures, I could move in next week.
Well, humans actually sent a few tardigrade astronauts on a mission to space in 2007 to see how tolerant we are against extraterrestrial-induced damage. It was discovered that our bodies activate both antioxidant defense and heat protection mechanisms to battle against the inhospitable conditions of outer space.
So there was no DNA damage to the astronauts?
Yep, so space adversities such as microgravity and radiation have no influence on our rate of survival.
Actually, we're still not fully sure whether that's true or not. There's also a possibility that the genomic defensive mechanisms repaired the astronauts' damage before they came back down to Earth.
We can basically do everything and anything we want with these genomic mechanisms in our body. No wonder we're thought to be pretty much immortal.
And this has also led to some useful genomic evolution byproducts, such as the tardigrade unique protein damage suppressor, also known as DSUP, which prevents radiation damage to our DNA.
DSUP? I've never heard of that protein before.
Well, it absorbs the radio damage through a strong electrostatic shielding that's formed from the positive amino acids and negative phosphates. This happens in intrinsically disordered proteins, also known as IDPs, which make up a major part of DSUP.
There's another way too. Since the DSUP is extremely flexible, it can form a complex DSUP DNA interaction by binding to the nucleosome to create highly tolerant structural support.
That's pretty cool. So how radio-tolerant are tardigrades in comparison to humans?
Well, the radio tolerance of an organism is inversely related to its genome size, and since we're near microscopic, we can expose ourselves to around 1,000 times the lethal radiation dose of humans and still walk out alive.
So overall, there's minimal DNA fragmentation happening in the tardigrade's body?
That's right.
You may be wondering now what kinds of experimentation this discovery inspired.
You read my mind. The damage suppression abilities are amazing. Wouldn't it be nice if we could just transfer the gene into other organisms and create superorganisms? That sounds too good to be true.
Surprisingly, that suggestion isn't too far-fetched. There have been multiple experiments in the past decade attempting to introduce the DSUP gene into organisms across the biosphere.
I heard about this. They began experimenting with human cultured cells and plant cells.
Wait, if it's only found in tardigrades, why would it be compatible with the nucleosome of completely different organisms?
Well, if we magnify the DNA of a tardigrade to the point where you could view the double helices, you can see that it has the same basic nuclear structure as any other living organism. That's the special thing about life on Earth. You can't tell the difference between organisms at extremely microscopic levels.
And the protein produced is also largely unstructured and disordered, allowing it to adjust to the shape of the bonded DNA.
So let me get this straight. Because DNA structure isn't an issue and the flexibility of DSUP allows it to form a DSUP DNA complex by binding to almost any nucleosome, the experiment should theoretically allow other kinds of cells to withstand damage from radiation exposure?
You're getting it?
OMG, no way. How successful were these experiments?
Well, starting with human cultured cells, irradiated cells were observed over several days, and the reproductive ability of cells without DSUP declined significantly more than those which were DSUP expressing.
By the fourth day, irradiated regular cells had an abnormal round shape and were detached from the culture dish, indicating cellular degeneration. Cells with DSUP retained their regular morphology for much longer. The proportion of fragmented DNA observed was reduced by approximately 40% in DSUP-expressing cells compared to regular cells.
Wow, can we use these findings yet? This could help patients undergoing chemotherapy.
Sadly, experimentation hasn't passed the stage of human culture cells, but there have been some amazing breakthroughs within plant cell experimentation.
What kind of breakthroughs?
Introduction of the DSUP gene to plant cells successfully produced an enhanced tobacco plant that grows under extreme radiation stress. Researchers ran a comparison of unaltered and altered plants where both were treated with ionizing radiation, UV radiation, and mutagenic chemicals.
How did they know the cells were successfully introduced with DSUP?
Successful integration was confirmed by examining the extracted target DNA and amplifying it using qPCR.
A higher concentration of DSUP protein was observed within the nuclear regions of the cell, protecting the nuclei from radiation damage. They were observed to grow better when exposed to genome mutagenic stresses over the unaltered plants.
That's amazing. Could this be implemented into crops to help them withstand future global warming crises related to excessive UV radiation exposure?
That's a possibility. Until further experimentation is conducted, we can't be 100%, but the implied possibilities are promising.
Wow, that was so interesting. I feel like I learnt so much about myself.
Yeah, it was great hanging out with you guys today, but I'll be going into anhydrobiosis for the next few years.
What a coincidence, so am I.
Aw, I'll miss you guys.
Thanks for listening. Tardigrade Trio signing off.